Yes, so today we are starting with the microbiology for the second year, which is the main subject. And starting with this, first of all, I give the knowledge of differential media and agar. So, what is the media? Culture media, agar, and various selective media, enriched media. These are various media for which an organism is mainly the bacteria. So, first, I will tell about the culture media. Culture media. Okay, so what is the culture media? It is the growth media for isolate the particular type of bacteria. Okay, so it is a growth media for isolation of that bacteria. So what are its constituents? What are the culture media is made up of? So basically there are three things mainly. First is the water, electrolyte, and third is peptone. Water is H2O, as we all know. The main electrolyte is NaCl, which you have to take in this, and the peptone, which is a partially digested protein. Okay, so it is a partially digested protein. Along with this, in the peptone, you can take extracts of meat, amino acid, riboflavin, and inorganic salts. Okay, inorganic salts. Inorganic salts, for example, potassium, magnesium, and phosphate. Okay, so I write down all the notes here and you can screenshot this slide. Yes, so the second one is agar. What is agar? Agar is basically the solidifying agent. It does not provide nutrition. ठीक है, अगार क्या है, एक सॉलिडिफाइंग एजेंट टाइप का है, पर ये कोई भी न्यूट्रिशन प्रोवाइड नहीं कर पाता. सो सॉलिडिफाइंग एजेंट, नो न्यूट्रिशन. ये बेसिकली मेरे नोट्स हैं जो मैंने बनाए थे और वही मैं आपके साथ शेयर कर रहा हूँ जो मैंने सेकेंड ईयर में अपने पढ़े थे, क्योंकि बहुत ज़्यादा कंफ्यूजिंग होता है. इट्स वेरी कंफ्यूजिंग टू रिवाइज ऑल ऑफ दीज नोट्स. सो आगे चलते हैं. अगर व्हाट इट सो अगर व्हाट आर इट्स कॉन्स्टिट्यूएंट एंड फर्स्ट इसमें हम लेंगे सीवीड्स. प्रोड्यूस कैसे होता है? सीवीड से. प्रोड्यूस फ्रॉम सी वीड्स, जो कि मेन आपका है रेड एल्गी. ठीक है, रेड एल्गी. सेकंड, जब इसको बनाना कैसे है वो है उसकी प्रोसेस बताऊंगा, हाउ टू मेक दिस अगार. सो फर्स्ट वी हैव टू टेक रेड एल्गी, देन वी हैव टू मेल्ट इट इन 98 डिग्री सेल्सियस एंड सॉलिडिफाई at 42 degree Celsius. Okay, then use 2% of it. 2%. एंड प्रिपरेशन के लिए हम इसको क्या करेंगे, इसमें पाउडर लेंगे हम. इन सब जितना भी ये अगार है इन सब का हम लेंगे पाउडर प्लस वाटर. ओके, एंड इन दोनों को हम मिक्स कर देंगे. वी हैव टू मिक्स दिस एंड sterilize it at in autoclave, basically autoclave in 42 degrees Celsius. Then we have to pour this in the petri dish and allow it to settle. So basically this is the agar. And third one is the meat extract. Meat extract is another way in which we can grow organism. So first is the beef extract, the main. The example of this is the lab lemco. Okay, and yeast extract is also there. So you can take both basically for the preparation. Okay, so in the third category we can discuss about the basal media. Okay, so basal media or simple media. Basal media or simple media. Okay, it's recording. Basal media or simple media, it is mainly used for the non-fastidious bacteria. Okay, non-fastidious bacteria in which we have to only have to grow that, only have to grow. Example, if we take example in this, first have we can take as a peptone water. Peptone water. So what is the peptone water? In the peptone water, it is 1% peptone plus NaCl, which is point and sorry, we have to take NaCl, which is 0.5% of it plus water. Okay, then second is the nutrient broth. Nutrient broth. Nutrient broth is made up of peptone water. Peptone means this peptone water comes here. Peptone water plus meat extract 1%. Okay, so third, you can see that in the second one we have to take peptone water plus add additional of meat extract. This is meat extract. Then in the third, which is nutrient agar, the important one, nutrient agar, in this we have to take this nutrient broth here along with 2% agar. Agar, what is it? Which I show you here. This is agar. We have to take this along with nutrient broth. It will form nutrient agar. And the fourth one, which is semi-solid media, which is semi-solid media. It is for the bacteria which can move or which have the motility. So basically used for the bacteria which can show motility bacteria. And in this for the preparation of this, we have to reduce concentration of agar to 0.2, say 0.5%. so it will form your semi-solid media. Okay, so now we have to discuss about the special mediums. So what are the first? The first one is enriched media. You have saw various enriched media. So what is enriched media? It is generally for the for only for growing pathogens in the medium. So only for growing of pathogens. Okay. Yes, so it is a culture media, basically culture media, and it is solid culture media. Okay, what it is made up of? It is made up of basal media. So basal media, which I have shown here, these are the basal media, any one of this plus enriched nutrients, so basal media plus enriched nutrients, we have to add enriched nutrients. Why? Because the pathogens which have to grow in this medium, they can get additional nutrients for adequate growth. So what are these enriched nutrients? These are basically blood serum or egg. Okay, so examples in this. So first has the blood agar, which you all know from your first day of microbiology class. In the blood agar, it can be enriched or differential. Okay, in blood agar, we can take the blood of horse or sheep for hemolysis. ठीक है, for hemolysis. Example of this blood agar. Sorry. Example of this, we can take like streptococcus pyogenes. Okay, streptococcus pyogenes and various other, but here I only take the one example as a classic example. In the second, which is chocolate agar, another example, chocolate agar. And what is the many of the students don't know what is chocolate agar and what is blood agar. Blood agar, we can see that we have to take horse and sheep blood for the hemolysis. And why we take chocolate agar? Chocolate agar, we can take, it is not because of the color, it is because that we have to take blood मतलब कैसे बनता है, आई विल शो दैट फॉर्मेशन। ब्लड लेंगे, इसको हम हीट कर देते हैं 70 डिग्री सेल्सियस पे। ओके, सो ये बन जाता है चॉकलेट अगर। क्लासिक एग्जांपल इसके निजेरिया एंड हीमोफिलस इन्फ्लुएंजा। ओके, सो थर्ड एग्जांपल, थर्ड classic example we have lofer serum slope. Lofer serum slope. The classic example have Corynebacterium diphtheriae, the most classic example. Okay, you can see that here is the serum. So next is the next enrichment media. Enrichment media. Okay, you can see we have enrichment media, and here what I take, enriched media. This is enriched media and this is enrichment media. So what is enrichment media? It is a liquid media. Okay, first point, it is a liquid media, and here we have to take basal media as a main base plus inhibitory agents. And in the enriched media we what we have take, basal media plus enriched nutrients for promote the growth of the particular bacteria. But here we take this inhibitory agents for what purpose? Because the purpose behind this for the inhibitory agent is for the promote growth of pathogens. Okay, pathogens, and not for the normal flora, and not for normal flora. ठीक है, the best classical example are the three. You have to remember these. First is the tetrathionate broth. Tetrathionate broth, which is for the Salmonella typhi. Okay, second one is the selenite F broth. Selenite F broth, sorry for my spelling mistake. Selenite F broth, it is used for the Shigella, GLLA. And the third one, which is the alkaline peptone water. Okay, which is for the Vibrio cholerae. So the next one कौन सा कलर लूँ मैं भाई? कौन सा, लाल ले लूँ? चलो भाई लाल ले लेते हैं फिर से. So next one is the selective media. What is the selective media? So देखो, जहाँ मुझे इंग्लिश यूज लगेगा मैं वहाँ करूँगा, और जहाँ हिंदी मुझे लगेगा, जहाँ समझाना होगा वो मैं कर दूंगा. ओके, so basically what we have is the next is selective media. It is the solid media plus inhibitory substance. हैंडराइटिंग मेरी थोड़ी माफ़ करना यार, थोड़ी मेरी गंदी हो सकती है. सॉलिड मीडिया plus inhibitory agent. Best example. Okay, best example I take in this, the Lowenstein Jensen medium. You have to remember this. Lowenstein, Jenson Jensen medium, used for this used for tuberculosis, Mycobacterium tuberculosis. Second is the TCBS. TCBS, the full form of TCBS is thiosulfate citrate bile salt sucrose. Okay, thiosulfate citrate bile salt sucrose, used for Vibrio cholera. Okay, you can take down my notes. Then third one is the DCA. What is DCA? Deoxy citrate agar. Okay, used for Salmonella and Shigella, used for both. Fourth example, XLD. These are the most important and the classic ones. This is the xylene, lysine, xylene, lysine deoxycholate. Then the fifth one is the potassium tellurite agar. Potassium tellurite agar, used for corynebacterium, diphtheria. Sixth example, most important, Wilson Blair medium, used for Salmonella typhi. Okay, so these are the six classic ones, which is Lovastein, TCBS, DCA, XLD, potassium tellurite agar, and Blair medium. Okay, so you can take down my notes here, and these are the knowledge about media and agar. So these are the base which you have to clear, and the next video is on the salmonella. Sorry, next video is on the staphylococcus aureus. Okay, so thank you.